thank you for giving me the opportunity to give this talk. So it's I'm going to present this work that I have done with Maria Cerveggio and Troy Day. So many organisms exhibit mate choice. So mate choice is composed of two components, giving the direction of preference. So, for example, does a female prefer the blue or the red male? And the choosiness, which gives the strength of a preference. So how much does a female prefer the red male over the blue one? So interestingly, there is empirical evidence that both preference and choosiness vary with age. So for example, here in roaches, the time of female response to male display that can be interpreted as a female choosiness decreases with age. So the general question I will be interested in this talk is why does choosiness vary with age? So there are actually very few theories on this question. So it's classically thought that because being choosy can decrease the chance to find a partner, it is often thought that choosiness should decrease with age. But actually, when doing a model with explicit age structure, it can be a bit more complicated. So, for example, here, Sozu and Seymour modeled changes in fecundity with age, in survival and extrinsic mortality with age, and they found that choosiness. So here it's just the inverse of choosiness, but so choosiness should first increase with age, reach a maximum, and then decrease with age again. So here I will address this question from a bit of a different point of view. So I will be interested in the interplay between age-specific choosiness and speciation. So mate choice is actually particularly important in speciation when there is a preference for mating partners from the same population or the same species. So when there is such a preference, strong choosiness will lead to reproductive isolation between populations, which is actually the most common criterion to define the species. So, however, there is a recent theory that showed that even if mating partners from the same species are preferred, like some moderate value of choosiness can evolve as an evolutionary optimum. And then, as a result, there would be some gene flow between well-defined species. So the assumptions behind this theory are that of a polygynous mating system where female choose males that have the same phenotype. So it's called phenotype matching. For example, red female would prefer red males, where alpha here measure choosiness. So I will use alpha for choosiness through the presentation. And so in this model, all females have the same mating success, but there is some variation in male mating success, as a male can mate with several females. So the model considers the scenario of a secondary contact between diverged populations. So the population, the population diverge in allopatry. So for example, one population becomes red and the other becomes blue. And then the population are connected again by some migration. So I will present this case in a bit more detail because I will use like all these results later on. So initially it was a model by Maria Cerveggio. So it's a population genetics model that considers that the mating traits depend on a single locus with two alleles, so say blue and red. And we are interested in the equilibrium frequency of at this locus in each population. So this figure shows the equilibrium lines for allele frequencies. So the solid lines show the stable equilibria, and populations are symmetrical. So that the top part is one population and the bottom part is the other population. So it's possible to show that divergence, so the difference in allele frequency between populations, is maximized at some intermediate choosiness value that is alpha opt, and that the system will evolve toward this choosiness value maximizing divergence, such that this choosiness value is an evolutionary optimum. So it's a stable evolutionary strategy. So why such intermediate choosiness evolves in this system? So there are several ways to see it, but one way is to consider that there is a trade-off on the mating success of the local male, so of the blue male in the blue population and the red males in the red population. So if when choosiness increases, the local males will have more mating opportunities with local females, and but less with the few migrant females. Whereas when choosiness decreases, the local males will mate more often with migrant females, and less with local females, because these females will also mate with migrant males. But so there is a choosiness value that actually maximizes the mating opportunities of the local males as compared to the migrant ones. So local males will benefit from both local female preference and have some mating opportunities with the migrant females. So then, when the mating success of the local males is maximized, the frequency of the local allele is also maximized, and population divergence is maximized. So how does this relate to the question of the evolution of age-specific choosiness? So, but if choosiness is very strong, so there is no question, choosiness should be strong at all ages. But if moderate choosiness evolves, there is several combinations of age-specific choosiness that could result in the same like moderate choosiness value over the lifetime. So it's also interesting to note that many species that are used for as a model species to study speciation have overlapping generations, and some of them have quite a long lifespan. And there is some empirical evidence that choosiness for partners of the same species change with age. So, for example, in mixed pairs of pied and collared flycatchers, it's often young females that are involved, but never old females. So the aim of the model will be to identify if speciation first can lead to the evolution of age-specific choosiness, and conversely, whether age-specific choosiness can affect the overall reproductive isolation between species, so can affect speciation. So to investigate this question, we developed a model of secondary contact, so very similar to the model of Maria that I just presented, where intermediate choosiness value is the evolutionary optimum. So the model considers an organism with overlapping generations and with several reproductive ages. So in this organism, there is one biallelic locus for the mating traits with allele big A and little a. So this locus can also be under variability selection. So the mating trait is called then a magic trait because it's both under sexual selection, so under mating preference, and under variability selection. So it's just that, for example, the blue individuals will survive better in the blue habitat, and the red individuals will survive better in the red habitat. And we also model a biallelic locus that is a modifier of age-specific choosiness, so with locus big M and little m. So this locus is not under variability selection, and evolution at this locus occurs only through linkage disequilibrium with the trait locus. So the mating system is again polygynous with a female choice and phenotype matching. And the female choosiness can then also vary with age. So this table here summarizes this assumption, where alpha x m, little m, is the choosiness of a female of age x and carrying the little m allele at the choosiness locus. And it shows that just a female carrying the mating trait big A will prefer males with the same traits, and similarly for little a females. So in this model, if choosiness doesn't vary with age, so the frequency of given genotype K in offspring depends on the frequency of each age class, so UX, on the frequency of female with age X and genotype I, on the frequency of male with genotype J, and on the probability that an encounter between so such a female and males will result in a mating. So this probability is actually given by the preference table, and on the probability that such mating will result in an offspring with genotype K. So it takes into account recombinations. So we already see in this equation that the effect of age just comes in through the age of a female. So this is because male is not a mating criterion, and it already suggests that any mechanism generating a difference in the trait frequency between males and females can, between males and a female of age X can affect the frequency of genotypes in the population and the evolution of choosiness. So the first step to understand the evolution of age-specific choosiness is first to understand how it affects divergence between population. So we first consider the version of the model where choosiness is fixed, so there is no polymorphism at the choosiness locus, for an organism with overlapping generation and two reproductive age classes. So first, we examined the case where there is no variability selection. So in this case, the frequency of the mating trait does not change with age. So it's shown in this little plot here. So the big plot shows the divergence. So measured as the frequency of a local allele as a function of choosiness. So the solid line shows the case where choosiness is not age-specific. So divergence in this case is maximized at some choosiness value, but we note again alpha opt. So it's shown by the vertical gray line. And the dashed line shows divergence as a function of choosiness at age 1, when choosiness at age 2 is fixed at alpha opt, And the dotted line shows the choosiness at edge 2 that maximizes divergence when choosiness at edge 1 is fixed at alpha opt. So this shows that without variability selection, there is a single choosiness value that does not depend on age, maximizing divergence. So now, if we consider a similar model with variability selection at edge 1, there is an increase in frequency of the locally adapted trait between edge 1 and 2 as a result of selection, which is shown here. Yeah, so here it's selection that generates this change in frequency, but other mechanism can generate such a change. So, for example, if there is some migration between edge 1 and edge 2. So again, the solid line shows divergence as a function of choosiness when choosiness is not age specific. So it's again maximized at some choosiness value alpha opt. And and this time, however, when we look at the choosiness at edge 1 that maximizes divergence when choosiness at edge 2 is fixed at alpha opt, the choosiness value is a bit lower. So it's the vertical dashed line. And if we look at the choosiness value at edge 2 that maximizes divergence, so the dotted line here, it's a bit larger than alpha opt. And we also see that divergence, maximum divergence is a bit larger when choosiness is age specific. So with variability selection, we see that age-specific choosiness maximizes divergence, and it seems to be related to this change in allele frequency between these two reproductive ages. So we try to have some insights on why we observe that. So there are several ways to understand it. So I propose you one. So we can consider the mating success of a big A male that we note XA with n females of age X, where the frequency of big A in this female is not PXA. So here again, the age of the male doesn't matter since age is not a mating criterion. So the mating success of this male is measured by the number of mating it can get with these n females, and it's a convolution of binomial of a mating success. So with each type of female, so the big A females and the little a females. So the expectation of the mating success of this male is simply proportional to the frequency of each type of females, weighted by the probability that this male, big A male, will mate with each type of female, with the two ratios. So so the mating success of this male again shows the trade-off between mating with a big A female and little a female, depending on choosiness. So it's possible to show that the choosiness value that maximizes the mating success of a big A male increases with the frequency of big A females. So as a result, if selection increases the frequency of local female, so say for example big A female in older age classes, the choosiness that will maximize the mating success of the big A males, so the local males, will also increase with age. So, of course, again, here I just use the age of female to put that into the context of an age-structured model, but it doesn't. The age here doesn't is not really necessary. Actually, any mechanism that affects the frequency of females will affect the choosiness value that will maximize the mating success of the chosen males. So, for example, it could be sex-specific selection. So then, to investigate the generality of these results regarding life history, so we simulated 1,000 variable life histories, again with 2 reproductive ages. So this plot shows the ratio of choosiness maximizing divergence at age 2 over age 1 as a function of the change in frequency induced by selection between age 2 and age 1. And there is a positive correlation between the increase in choosiness with age and the effect of selection on the change in frequency of a locally adapted allele. So it's interesting to see that the change in choosiness between age 1 and 2 can be up to threefold. So then if we look at the population scale, so the question is to see if the mean choosiness in the age-structured model is different from the choosiness that maximizes divergence when it is not age-specific. And so to do that, we simply compare both values, and we find that it is the same. So the reproductive isolation is not affected by modeling explicitly age structure. So a similar approach shows that the level of divergence between a model with age-specific choosiness and a model without age-specific choosiness is similar, at least at the big scale. But when we look a bit more closely, we find this distribution for the difference in divergence between both models. So divergence is actually a bit larger in a model with age structure as compared to a model without age structure. So overall, even though there is a large variation in choosiness with age maximizes divergence, at the overall, the age-specific choosiness has little effect on speciation in the sense of gene flow between populations. So now that we showed that age-specific choosiness maximizes divergence when there is variability selection, and that we got some insight of why, the question is whether such age-specific choosiness values maximizing divergence would actually evolve. So to address this question, we built a pairwise invisibility plot where we test whether rare mutants can, so a rare mutant coding for some choosiness value could invade a population with some resident choosiness. And the black area shows the combination of resident choosiness that can be invaded by some mutants. So then first we consider the case where choosiness is not age-specific. So the white line here shows an evolutionary succession. So it leads to some singular choosiness value. So this choosiness value is called evolutionary stable because it cannot invaded by any mutant. And the red lines shows the choosiness value maximizing divergence. So it is alpha opt that we had before. And so there is a correspondence between the evolutionary stable choosiness value and the choosiness value maximizing divergence. So alpha opt here is also an evolutionary stable choosiness strategy. So now again, we consider the case where the mutant affects only choosiness at age 1, while choosiness at age 2 is fixed at alpha opt. And we have the same pattern. So, so the red lines show the choosiness value at H2 that maximizing divergence. So it's a bit lower than alpha opt. And it's again an evolutionary stable strategy. And similarly, when we look at choosiness at H2, we find that the choosiness at H2 that maximizes divergence is evolutionary stable. So overall, we found that age-specific choosiness that maximizes divergence is an evolutionary stable strategy. So we actually used several other methods to confirm these results because we couldn't demonstrate it formally. So it's with just a combination of numerical methods. And for example, we tested the fate. So we tested directly the fate of mutants coding for choosiness value different from the age-specific choosiness values maximizing divergence. So the black line here, for example, shows the fate of a mutant affecting choosiness at age 1, and the red lines shows the fate of a mutant affecting choosiness at age 2. And in all cases we tested, the mutant always decreased in frequency as compared to the choosiness, the age-specific choosiness values maximizing divergence. So yes, I will just conclude on this model. So the difference in frequency of the mating traits between partners affects the evolution of choosiness. So it's actually a pretty general results, but go a bit beyond the pure case of an age-structured model. So in the particular case of an age-structured model, viability selection increases the frequency of a locally adapted trait with age, which can induce an increase in choosiness with age. But this effect, this effect within the life history does not affect speciation at the larger population scale. And there is overall more theory needed to investigate a specific variation in mate choice and the implications it can have on speciation and adaptation. So another interesting thing in this model is that it can generate an apparent preference for all males just because variability selection so will again increase the frequency of a mating trait with age. And if females prefer the locally adapted allele, they are going to mate more often as a correlation with old males. So actually, so this is interesting because there has been a lot of theory trying to understand why there is this apparent preference of females for old males in nature. So here we show that it can be can be just an artifact of other mating rules. And also we we saw that the fitness surface around the ESS is relatively flat. So, for example, here this is what PIP will look like after 20,000 generations, and this question about whether selection can actually optimize choosiness at all ages. So we started to have some general analysis on this problem, but this is a problem that also deserves more investigation. I will, so I finish here, and I thank you for your attention.
Thank you, Olivier. Are there any questions? Okay.
Hi, Olivier. It's Vincent Calvet speaking. If I understand well, your conclusions are based on one specific scenario where you have two populations that already diverged in the past, and that come again in contact. Is it the case?
Yes, it could be, but what's important in the model is very viability selection. So there must be some viability selection that maintains population divergence, and that affects the frequency of mating trait within a generation.
Okay, so my question was how robust are the conclusions with respect to the scenario, but what you say is that the scenario is not so important that what it implies for the population.
Yes, so the model is we focused on the case of an age-structured population, but so we found that the increase in that selection generates a difference in frequency between the female that make the choice because and the males. So because the male age is not a mating criterion, all the males are considered as like a single mating pool, whereas when we are interested in age-specific choosiness, what matters is really the frequency of the mating trait within the cohort of choosy females of a given age. But this difference in frequency between like all the possible males and a given group of females can be generated by other evolutionary forces than just viability selection. It could be sex-specific selection. So if there is some selection on females but is not on males, it generates a difference in frequency between females and males, which can affect the evolution of choosiness in females. It could be migration. It could be other forces. So it's just an open door for further research on this the evolution of choosiness in general.
Thank you very much.
Okay, another question. Yeah.
Hi, thank you very much for your talk. It was very interesting, among other things, because it reminded us that it's the females who choose and not the males, because it's the females who have the power and not the males. So my questions are twofold. Indeed, I think you are interested in the age variation of choosiness. Maybe the criterion of choices could be more biologically, but I'm a mathematician, so I don't know, maybe it's stupid. But maybe the criterion of what they choose on would be more biologically relevant. For instance, I remember reading some articles that said that for a species which I love, especially the humans, well, so in some countries, some areas of on the Earth, the females have decided that the males would be all black hair, for instance. And it seems to have been necessarily through the sexual selection, which is the Darwinian concept, of course. So do you think that the dependence on age of choosiness is the most relevant parameter for the speciation, which is, by the way, well, the species, you have written a preference for the same species. Well, if there's a preference for another species, by definition, there will be no offspring. So what is do you think the age dependence of choosiness to be the most relevant parameter?
So first, it's not because there is, so preference just gives a probability of mating. And so preference gives a direction of a preference, but choosiness gives the probability that the mating will happen with preferred partners. So it doesn't exclude that there is a mating with other species. And actually, we see that like different species can mate together. And we also, so there is also quite a lot of empirical evidence that choosiness varies with age. So it means that there are mutations affecting age-specific choosiness. And actually, there is variation in choosiness with age, and actually this variation in choosiness and even sometimes in preference with age is very poorly understood from a theoretical point of view. So yeah, we just propose just some theory to help understand the pattern we actually observe.
Thanks. Yeah, maybe a quick question.
Thank you for this nice talk. My question was, so you assume here that choosiness is encoded by a single locus or a modifier, but if you think that it could be polygenic, how would that sort of affect your patterns?
Okay, so
I'm just asking for like an intuition, not like a very precise.
Yeah, so my intuition is that what we are really interested in is an optimal choosiness value, and I think the way it is encoded doesn't really matter because it's more like we are just looking at a strategy of mating, and in the end, like the precise determinism of doesn't really matter. However, I think it can change considerably the dynamics of to reach the equilibrium. But it would be my intuition, more on the dynamics than on the final equilibrium strategy.
And for example, on speciation, do you think it would improve the chance that it happens, or?
Yeah, I have not much intuition on that.
Okay.
Yeah, I don't know. Honestly, I don't know.
Thank you.
Okay, thanks. Other questions? Yeah. Okay.
Thanks. Hi, Olivier. This is Denis Rose. I had just a general question on how you define divergence, because the way I understood it is that here that it is, you talk about divergence between the two populations, right? But not divergence between two genetic entities, because otherwise I would expect that by maximizing choosiness, you would maximize divergence, right?
Yes, I'm looking at divergence is measured as really as the frequency of locally adapted traits in here. I don't know if it's clear. It's as the population scale.
So the link with speciation is not completely clear to me, because for me, so the fact that you maximize divergence, the way you define divergence, doesn't necessarily mean that it will maximize reproductive isolation, right?
No, yeah. So the point here is actually that there are actually quite many cases where selection doesn't push toward the evolution of more choosiness, like the optimum for maximizing the optimum for maximizing sexual selection is not by increasing choosiness, is at some intermediate choosiness value. And if the choosiness locus is not under other selective force, then choosiness will be stopped at some optimal choosiness value corresponding to a maximum in sexual selection, in the mating, in the variance in mating success of the males.
Okay, thank you.
Okay, thank you, Olivier. So I think we're done with questions. So we may thank all the speakers for this day. And thank you, Olivier.
Thank you.